Yes, good day, my dear students. Welcome to another section of BCH 407, lecture 4 on the series. The title is Nature of Drugs and Transport Across Membrane. But before we go into it, I want to refresh our understanding of lecture 3, wherein we define and explain the aims of chemotherapy, which is actually curative, disease control, and palliative. And we actually explained why rapidly dividing cells are differentially sensitive to anti-cancer agents. In that lecture, we review the cell cycle, the G1S and G2 and M phase, and how many drugs are cell-specific and the ones that are non-cell specific. We also went ahead to talk about the major classes of anti-cancer drugs and summarize their mechanisms of action. The lecture also covers molecular mechanism of drug resistance, and I hope you have taken time to listen and follow the lecture as it demands. Today, this is lecture 4, and our focus will be on nature of drugs and transport across membrane. Nature of drugs and transport across membrane. Now, what are we expected to achieve at the end of this lecture? We have 5 points. 1. You should be able to describe drug, its basic chemical and physical properties, explain the structure and selective permeability of biological membranes, describe the major mechanisms of drug transport across membranes, differentiate between passive diffusion, facilitated diffusion, active transport, and vesicular transport. Finally, you should be able to identify the factors that affect drug transport in the body. Now, we are starting with chemical and physical properties of drugs. A drug possesses specific chemical and physical properties or characteristics that determine how they behave in the body. The chemical properties of drugs, which are examined, include chemical composition, molecular structure, functional groups, stereochemistry or optical isomerism, acid-base properties or ionization, then the chemical stability. Now let's examine these ones one after the other, starting with chemical composition. Now, organic drugs contain carbon and form the majority of therapeutic agents. Examples include our popular paracetamol, aspirin, morphine that are used to reduce pain. Inorganic drugs are mainly mineral-based. Examples are ferrous sulfate, magnesium sulfate, and so on and so forth. Now, The molecular structure is simply an arrangement of the atoms in a drug which determines its interaction with receptors. Very soon, we shall have an in-depth discussion on receptors. Now, the functional groups, they determine whether a drug behaves as an acid, base, or neutral compound. They influence solubility, reactivity, binding to biological targets. Now, stereochemistry. Some drugs exist as mirror image of itself, which we refer to enantiomers. In this case, we have the L and the D form. One form is more active than the other. The D forms are actually more active than the L forms. Now, the acid-base properties or an ionization. Most drugs behave as weak acid or weak bases, where their ionization depends on pH and pKa. Also, non-ionized forms of drugs cross membranes more easily. This affects absorption and distribution. Now, the chemical stability of drug. Refers to the resistance to chemical breakdown, and drugs may degrade as a result of heat, light, oxygen, and moisture. Of course, you will note that unstable drugs lose potency over time. Now, let's look at the physical properties of drugs. Drug physical properties refer to the observable and measurable features that influence formulation and absorption. These are solubility, physical state, particle size, crystalline and amorphous forms, polymorphism, melting point, hydroscopicity, and organoleptic properties. Now, solubility of a drug is the ability of a drug to dissolve in solvents, have good membrane penetration, requires lipid solubility. What I mean by that is that good membrane penetration of a drug requires lipid solubility. Good circulation in blood requires water solubility. And the physical state affects handling and administration. So, drugs may be water soluble, which is hydrophilic, lipid soluble, lipophilic, and they may exist as solid, liquid, or gases. Solid as you have in tablets, capsules, and powders, liquids, syrup, and injections, gases, anesthetic gases, the one that is employed during operational procedure or surgical procedures. Particle size, crystalline and amorphous form. Smaller particles dissolve faster. Faster dissolution leads to quicker absorption. Of course, you can imagine that. Crystalline drugs have regular structure and are more stable. Amorphous drugs are irregular and dissolve faster but are less stable. Please note the differences. We will combine polymorphism and melting point together. Polymorphism in drugs refers to a situation where some drug exists in more than one crystalline form. Different forms may have different solubility and bioavailability. Melting points indicate purity and stability. High melting points usually means strong intermolecular forces, and this affects formulation and storage. Hydroscopicity and organoleptic properties. Considering first hydroscopicity, some drugs absorb moisture from air, which may lead to degradation or clumping. For such drugs, they require special packing or packaging. On the other hand, organoleptic properties refers to taste, color, odor, and texture of a drug. Briefly, let us look at the structure of membrane, which you have done in BCH 303 in membrane biochemistry. Now let's just briefly have a recap of the membrane structure. The picture before you is a simplified diagram of the membrane structure and its components. Of course, you know that the cell membrane acts as a protective barrier and a regulatory gateway. Its selective permeability ensures that substances enter the cell, waste products or other harmful substances are barricaded from entering the cell. Now to selective permeability of membranes. What are permeable substances. Permeable substances to the cell membrane are small, non-polar, lipid-soluble molecules such as oxygen, carbon dioxide, and some drugs that can easily pass through the membrane by diffusion. On the other hand, we also talk about partially permeable substances. The partially permeable substances are small polar substances or molecules like water, urea, that can slowly pass through special channels. So, in essence, selective permeability refers to the ability of the cell membrane to allow some substances to pass through while preventing others. Impermeable substances, these are large molecules and charged ions that cannot cross freely but require specific transport proteins. Now, the selective permeability is controlled by the lipid nature of the membrane, the size and charge of the molecules, and the presence of transport proteins. I'm sure you are already familiar with these. Now, to the mechanism of drug transport across membranes. First, we have passive diffusion. This is the most common mechanism of drug transport. This is the most common mechanism of drug transport across membrane. The features are that the drug moves from a high concentration to a lower concentration region. No energy is required. No energy in the form of ATP is required, and it does not involve carrier proteins. Second is facilitated diffusion. This method involves the use of carrier proteins to assist the drug movement. The features of facilitated diffusion include movements along a concentration gradient. Again, no energy is required. It's mediated by carrier protein, and it is saturable because it has a limited number of carriers. The third is active transport. Active transport moves drug against the concentration gradients. Remember the first one, it moves from a point of higher concentration to a region of lower concentration. But in this case, the active transport moves drug against the concentration gradients. In this case, it requires ATP, energy in the form of ATP, to drive that movement. Carrier protein is required. It is selective and saturable and can be inhibited by metabolites. Still on mechanism of drug transport across membrane, we talk of filtration or bulk flow. This is movement through membrane pores due to pressure differences. The features are that it depends on hydrostatic pressure. No energy is required and it is not selective. This method is suitable for water-soluble molecules. Now, still on method of drug transport across membrane, we talk about ion pair transport. This is, in this mechanism, charged drug combine with oppositely charged ions to form neutral complexes. The feature include neutral complexes, cross membrane easily. That is, the neutral complexes that are formed cross the cell membrane easily. Now, after crossing the cell membrane, they dissociate into their individual components. Now, the importance of this is that it enhances absorption of highly ionized drug. So drugs that are highly ionized pass through cell membrane through this method. Now, endocytosis and exocytosis. Now, this method involves transport using membrane-bound vesicles. Endocytosis and exocytosis, where we have the endocytosis. Here, the cell membrane engulfs substances and form vesicles within the cell. The types include pinocytosis, for fluids, receptor-mediated endocytosis, specific for molecules, while exocytosis involve the release of substances that have been engulfed by the cell. Now, the endocytosis and exocytosis exemplifies the vesicular transport method or mechanism. Now, we have talked about transport, nature of drugs, physical and chemical properties. What are the factors that can affect drug transport? They are lipid solubility, degree of ionization as mentioned, molecular size as mentioned, membrane thickness, blood flow, presence of transport proteins or carrier proteins, then pH of body fluids. All these are factors that determines the success or failure of drug transport or in drug transport. Now, having said that, let us summarize what we have listened to. We have heard about chemical and physical properties of drug, structure and selective permeability of biological membranes, major mechanisms or methods of drug transport in the body, and these include passive diffusion, facilitated diffusion, active transport, filtration, ion pair transport, and vesicular transport. We concluded by giving factors that act as biological barriers to drug movements within the body. As usual, I have provided a set of study questions which you can use to assess your comprehension of this lecture. Take your time, go through them, appraise and tease yourself with them in order to rate your own understanding of what has been presented. As usual, do your group discussion, consult your reference text for clarity and better understanding of what has been presented now. In the nearest time, we'll try to fix a physical class for interaction to clear all gray areas. Thank you. This is the end of lecture number 4.